Good evening, everybody. I'm Bobby back with you. Today we'll be discussing allosteric enzymes as part of the discussion on the first chapter on molecules in life sciences and related aspects in Neet Biology. So stay tuned. I'll be back with you in a moment, right?
Right. I'm back with you again. Today we're continuing on the Michaelis-Menten model. So before I can proceed, let me just give you a little explanation about what this Michelin-Menten model is about. See, it's actually a model of enzyme kinetics, which explains how the rate of an enzyme catalyzed reaction depends on the concentration of the enzyme and its substrate. Now look here. Now, if we look at the Michaelis-Menten method, you can see that the Michaelis-Menten equation is the rate equation for a one substrate enzyme catalyzed reaction. This equation relates the initial reaction rate VO, the maximum reaction rate Vmax, and the initial substrate concentration through the Michaelis constant Km, a measure of the substrate binding affinity. Right? And other aspects like what is the significance of the Michaelis-Menten model? Examining enzyme kinetics is critical for understanding cellular mechanisms, systems, and for using enzymes in industry. This Michaelis-Menten equation has been widely used for over a century to estimate the enzyme kinetic parameters from reaction progress curves of substrates, right? And what is the concept of Km and Vmax in Michaelis-Menten equation? So I'm just giving some question answers so you have some explanation to what's being taught and you know where you stand as well, right? So Vmax is the maximum reaction velocity at which all enzymes become saturated with substrate Km, right? It's Km is a substrate concentration at which half of the velocity, maximum velocity, is achieved. We can determine Vmax and Km for a reaction by plotting a graph of the rate of reaction V against the concentration of the substrate S. The three assumptions of Michaelis-Menten. Three assumptions are implicit in Michaelis-Menten kinetics: the steady state approximation, the free ligand approximation, and the rapid equilibrium approximation. The Briggs-Haldane approach frees us from the last of these three. And what is the Michaelis-Menten plot used for? The graph can be used to calculate the Michaelis constant Km, which is the concentration of a substrate required in order for an enzyme to act. The Michaelis-Menten equation has been widely used for over a century to estimate the enzyme kinetic parameters for the reaction progress curves of substrates. Right? And what are the principles of Michaelis-Menten? Michaelis and Menten assumed that the substrate binding and dissociation occurred much more rapidly than product formation, kcat, much less than k of the rapid equilibrium approximation, and that therefore the Km will be very close to the Kd. The larger the kcat is relative to k of, the greater the difference between Kd and Km. Why is Michaelis-Menten better? Michaelis-Menten plot. Okay? But it remains of major interest because it has another valuable property. It allows the properties of isoenzymes catalyzing the same reaction, but active in very different stages of substrate concentration to be compared on a single plot. The applications of Michaelis-Menten kinetics. Apart from biochemical reactions, it has been applied to a wide variety of other areas such as alveolar dust clearance, clearance of blood alcohol, bacteriophage infection, and photosynthesis irradiation relationships, right? And how is the Michaelis-Menten constant used? Substrate affinity and the apparent Michaelis-Menten constant, the affinity of an enzyme for a substrate, is commonly measured using the apparent Michaelis-Menten constant Km, which can be defined most simply as the amount of substrate necessary to allow an enzyme to function at half its maximum velocity. And what is the function of Michaelis-Menten kinetics? The enzyme E and the substrate S. First combines to give an enzyme substrate complex ES. Then the chemical processes take place in a second step to break down ES and produce product P with the first order catalytic constant Kcat, also called K2, or the turnover number. How is the Michaelis-Menten equation derived? The Michaelis-Menten equation arrives from the general equation for an enzymatic reaction E plus S given equal to EH, again in reverse of 2, E and P, where E is the enzymes, S is the substrate, ES is the enzyme substrate complex, and P is the product. What is the full form of Vmax? Vmax, or maximum velocity of an enzymatic reaction, can be defined as the rate of the reaction at which the enzyme shows highest turnover. What is the unit of Km in Michaelis-Menten? Km is the Michaelis-Menten constant. In the same unit as X, it is the substrate concentration needed to achieve a half maximum enzyme velocity. What does V stand for in Michaelis-Menten equation? Michaelis-Menten equation. In this equation, V0 is the initial velocity of the reaction. Vmax is the maximum rate of the reaction. The variable V is also referred to as the rate of catalysis of an enzyme. For different enzymes, V varies with the concentration of the substrate S. And what are the steps of the Michaelis-Menten model? Michaelis-Menten model for enzyme kinetics presumes a simple two-step reaction. Step 1 is binding. The substrates binds to the enzyme. Step 2, catalysis, The substrate is converted to product and released. Note that enzymes not matching this reaction schema still show similar kinetics. What are the limitations of Michaelis-Menten? The Michaelis-Menten equation does not apply when a substrate is inhibited or activated as a result of a second substrate molecule being bound. And the Michaelis constant Km, or the substrate concentration at a half maximum velocity, does not equate to the affinity of the enzyme for its substrate. And what is the Michaelis-Menten constant Km equal to? Michaelis-Menten constant Km is equal to substrate concentration at which the rate of reaction is maximum, the rate of reaction, the rate of enzymatic activity. Can Km value be negative? Even though the Km value is negative, its reciprocal is positive, which is why it appears as a positive number on the Lineweaver-Burke plot. So why is Km negative for Lineweaver-Burke plot? Burke plot hint we BrainLeader in has an explanation for us. Let's see what it has to say. I'm afraid it's not clear there. But then we have a little bit of an explanation. V by Vmax is equal to S by Km plus 1 by Vmax, where V is the reaction velocity, S is the substrate concentration, Km is the Michaelis constant. Vmax is the maximum velocity, reaction velocity. Lineweaver bug plot is created by plotting 1 by v versus 1 by s. When this is done, the slope of the line is equal to minus 1 by km. And the y-intercept is equal to 1 by Vmax. The Km value is the substrate concentration of which the reaction velocity is half of Vmax. Because the Km value is in the denominator of the equation, it is negative on the Lineweaver bug plot. The reason for this is that the y-axis of the Lineweaver bug plot is 1 by V, which is the reciprocal of V. When you take the reciprocal of a negative number, as a result is a positive number. So even though the Km value is negative, its reciprocal is positive, which is why it appears as a positive number on the Lineweaver bug plot, right? So we're going back to our subject proper, where we stopped, right? Okay, we're back with the subject proper. We stopped here, and now, in fact, for many enzymes, experimental evidence suggests that Km provides an approximation of substrate concentration in vivo. When the Km is known, the fraction of sites filled FES, enzyme substrate, which I just now explained to you, any substrate concentration can be calculated from Fe is equal to V by Vmax is equal to substrate by substrate plus Michelin constant. So the second Km is related to the rate constant of the individual steps in the catalytic scheme. Right, and Km is defined as K minus 1 plus K2 by K1. Consider a limiting case in which K minus 1 is much greater than K2. Under such circumstances, the ES complex dissociates to E and S much more rapidly than product is formed. Under these conditions, K minus 1 is greater than or much greater than K2. The dissociation constant of the ES complex is given by Km is equal to K minus 1 by K1. Okay, ES is equal to ES by ES, so K minus 1 by K1. In other words, the Km is equal to the dissociation constant of the ES complex if K2 is much smaller than K minus 1. When this condition is met, Km is a measure of the strength of the ES complex, enzyme substrate complex. A high Km indicates weak binding, a low Km indicates strong binding. It must be stressed that Km indicates affinity of the ES complex only when K minus 1 is much greater than K2. The maximal rate Vmax reveals the turnover number of an enzyme, which is the number of substrate molecules converted into product by an enzyme molecule in a unit time when the enzyme is fully saturated with the substrate. It is equal to the kinetic constant K2, which is also called Kcan. The maximal rate Vmax reveals the turnover number of an enzyme with the concentration of active sites, e.g. is known because K2 is equal to Vmax by ET and Vmax is equal to K2 by ET. I'm just going into the subject matter proper with the various figures on exhibition. You can see them. And continuing here with the subject, we are almost closing down on this topic. So in other words, the slope of the plot is increased by the Michelin-Menten plot is increased by the factor 1 plus 1 by k in the presence of a competitive inhibitor. In non-competitive inhibition, the inhibitor can combine with either the enzyme or the enzyme substrate complex. In pure or non-competitive inhibition, the values of the dissociation constants of the inhibitor and enzyme of the inhibitor and enzyme substrate complex are equal. The value at Vmax is decreased to a new value called V approximate max. Vap max, and so the intercept on the vertical axis is increased. The new slope, which is equal to Km by Vap max, is larger by the same factor. In contrast, the Vmax Km is not affected by pure non-competitive inhibition. The maximal velocity in the presence of a pure non-competitive inhibitor Vmax is given by Vap max is equal to Vmax by 1 plus 1 I by Ki. So that's it about that topic there. Next is allosteric enzymes. The Michelin-Menten model has greatly assisted in the development of enzyme chemistry. Its virtues are simplicity and broad applicability. I just now read those details for you. However, the Michaelis-Menten model cannot account for the kinetic properties of many enzymes. That is a drawback. An important group of enzymes that do not obey Michaelis-Menten kinetics comprises the allosteric enzymes. These enzymes consist of multiple subunits and multiple activity sites. Drawback of the Michaelis-Menten model. The allosteric enzymes have an allosteric site in addition to the active site. Allosteric is derived from the Greek word allo and steric is other and steric is at other sites. An effective molecule can bind with this site and can either activate or deactivate the enzyme molecule. The effector molecule is termed as modulator, regulator, or ligand. The effector molecule may be either activators or inhibitors. The activators are generally one of the substrate of the cofactors. In most cases, allosteric inhibitors are the end products of the metabolic pathways in which particular enzyme is participating. Thus, this kind of enzyme inhibition is also called as feedback inhibition or end product inhibition or retro inhibition. The effect of allosteric is reversible. When they are withdrawn, the enzyme resumes its original activity. Allosteric enzymes often display sigmoidal plots of the reaction velocity V0 versus substrate concentration S, rather than the hyperbolic plots predicted by the Michaelis-Menten equation. Two models explain the kinetic behavior of allosteric enzymes. The sigmoidal dependence of VO on S reflects subunits cooperatively and has inspired two models to explain these cooperative interactions. The first model, the symmetry model proposed by Jacques Monod and colleagues in 65, allosteric enzyme can exist only in two conformations, active and inactive. All subunits are in the active form or are all active. Every substrate molecule that binds increases the probability of a transition from the inactive to the active state. Now, as a continuation, I'd like to give you a little bit of explanation about bioenergetics and its significance. So basically, bioenergetics is a subfield of biochemistry that investigates how cellular processes convert different forms of energy, most frequently through the production, storage, or consumption of adenosine triphosphate ADP. The majority components of cellular metabolism and hence life itself are dependent on bioenergetic processes. And some examples of these activities are cellular respiration and photosynthesis. Now, bioenergetic studies energy flow in living systems. This is an active area of biological research that includes a study of energy transformation in living organisms and thousands of different cellular processes, such as cellular respiration, metabolic and enzymatic processes, and later production and use of adenosine triphosphate ATP molecules. Bioenergetics describes how organisms acquire and transform energy to perform biological work. Bioenergetics studies metabolic pathways. And continuing there, bioenergetics studies the energy required to form and break chemical bonds in biological molecules. It also studies the study of various energy relationships in living organisms. The ability to harness energy from a variety of metabolic pathways is a property of all living organisms that contain the earth signs. Growth, development, anabolism, and catabolism are some of the central processes in the study of biological organisms. Now, some organisms, such as autotrophs, can acquire energy through sunlight, through photosynthesis, without needing to consume nutrients and break them down. Organisms like heterotrophs must take nutrients from food to be able to sustain energy by breaking down chemical bonds and nutrients during metabolic processes such as the glycolysis and the citric acid cycle. So we've studied all this so far. Importantly, as a direct consequence of the first law of thermodynamics, autotrophs and heterotrophs participate in a universal metabolic network. By eating autotrophs, plants, heterotrophs harness energy that was initially transformed by the plant during photosynthesis. So we have an explanation of what we've been studying so far. In a living organism, chemical bonds are broken and made as part of the exchange and transformation of energy, which is available for work such as mechanical work, other processes such as chemical synthesis and anabolic processes and growth. Adenosine triphosphate is the main energy currency for organisms. Metabolic and catabolic processes create ADP from amine and static materials and break it down into ADP and inorganic phosphate for use in biological functions. Cell energy charge is a ratio of ATP to ATP concentrations. If there is more ATP than ADP, the cell may utilize ATP to conduct work, otherwise it must synthesize ATP by oxidative phosphorylation. Through oxidative phosphorylation, living organisms make ATP from sunlight or O2. ATP's terminal phosphate bonds are weak compared to those produced when ATP is hydrolyzed to adenosine diphosphate and inorganic phosphate. The phosphoanhydride link between term phosphate group and the remainder of ATP does not contain this energy. ATP stores energy in cells. Every living creature uses chemical energy for molecule bonds rearrangements. And reactions. Exergonic reactions release energy spontaneously. Negative G indicates thermodynamically favorability. Gibbs free energy. Activation energy moves reactants from a stable state to a high energetically unstable transition state to a more stable low energy state. Complex reactants break into simpler products, usually catabolic. Because reactants have more energy than products, Gibbs free energy is negative G. Alright, and a chemical process that consumes energy is called endergonic. Opposite of exergonic, it has a positive G because H is greater than 0, which indicates it takes more energy to break reactants bonds than product bonds. That is, products have weaker bonds than reactants. Endergonic reactions are thermodynamically favorable and won't occur at constant temperature. Endergonic reactions are anabolic. G is equal to HTS, where G is equal to Gibbs free energy change, H is equal to enthalpy change, T equal to temperature in kelvins, and S is equal to entropy change. Right? Examples of bioenergetic processes are glycolysis, where it breaks down glucose into pyruvate, yielding 2 ATP per glucose molecule. When a cell has more ATP than ATP, that is high energy charge, it cannot execute glycolysis to release energy from glucose. Pyruvate is a glycolysis product that may shuttle into other metabolic processes, gluconeogenesis, etc. Glycolysis also creates NADH, which donates electrons to the electron transport chain. When the cell's energy charge is low, the concentration ADP is larger than that of ATP. It synthesizes glucose from carbon-containing molecules such as proteins, amino acids, lipids, pyruvate, etc. Proteins may be broken down into amino acids, which are needed to make glucose. The citric acid cycle, acyl coenzyme A is initially reacted with oxaloacetate to generate citrate. Either processes yield carbon-containing metabolites. These metabolites are sequentially oxidized, and the oxidation energy is preserved as FADH and 2 and NADH. When the transmit electrons to the electron transport chain, these reduced electron carriers are reoxidized. Ketosis uses ketone bodies as well as cell energy instead of using glucose. When glucose levels are low, such during hunger, cells switch to ketosis for energy. The next is oxidative phosphorylation releases energy from O2's weak double bonds in the electron transport chain. NADPH, FADH2, and NADH contribute electrons to redox processes in electron transport chain complexes. Redox reactions occur in mitochondrial enzyme complexes. These processes carry electrons down the proton-derived electron transport chain, and the difference in proton concentration drives ATP production via ATP synthase. Photosynthesis uses sun energy to generate glucose from carbon dioxide and water in plants. Chloroplasts respond after synthesizing glucose. Plant cells can create ATP by photophosphorylation. Human vitamin D production results in ATP. And the conclusion, the field of biochemistry known as bioenergetics studies how cells use adenosine triphosphate ADP to store, produce, and release energy, ATP. The majority of cells' metabolism, cellular metabolism, and by extension of all life depends on the bioenergetic activities like cellular respiration and photosynthesis, right? As a continuation of this subject, we are extending into and relating our studies of life sciences and the NET section, CSIR NET, and concomitantly we are less detailing but trying to focus on studies that relate to the same in cell biology for the Neet category, where you have the topics of tools and techniques in cytology, cell as a unit of life, biomembrane, structural organization of cell, biomolecules, enzymes, cellular metabolism, and cell reproduction. In this, we have the subject of biomolecules and the structural organization of cells, enzymes, and cellular metabolism. So we're dealing with cellular metabolism. So what I'm speaking to you now has a lot of significance in cell biology. So we're going over to page number